Right, so we are now being recorded. So again, welcome to our seventh video teleconference for BIOL 105L YB. So we have not met for almost a month because of the midterms week. Um, but now we are back on track, and in this video teleconference, the agenda that I set would be to review on the previous laboratory exercises. So we will not anymore go back to the midterm lab exercise. Okay, so I think we ended with the SDS page, if I'm not mistaken. So in this review, we're going to start from DNA extraction. That's exercise number 8. So if you look at our calendar, we are actually now currently in sync with the number of video teleconferences that we're supposed to have. This is the 7th, and exactly in the calendar it says the 7th. So that's good. And currently right now you're supposed to be in the middle of making your lab reports for exercise 8, and the deadline for that will be this upcoming Saturday. Okay, so if you look at the schedule ahead, we have one more exercise, and then after that will be the semifinals for this particular subject. So again, these are the exercises that we had done already, including this week. Exercises 7, 8, and then we have exercise 9. Now, these three exercises are actually part of the same workflow in the laboratory, but they are separated into three to make it more manageable, no, and easier to keep track of the steps. But in reality, if you are working in the laboratory, when you do DNA isolation, it doesn't end with just DNA isolation. You do something with the DNA. And that's what we did in exercise 8 in the polymerase chain reaction. And the product of PCR is also not the final. So the last part of the workflow will be exercise 9 to be done next week, and that's agarose gel electrophoresis. So for this afternoon, let us review what were done in exercises 7 and 8. And I'm also going to explain some of the important points on those processes, now especially PCR. So please also speak up if I ask you to give your opinions and to recite, because I don't want to be speaking all the time, because we already did this. You can already relate to what is happening here. Right, so let's start. So first, exercise number 7, DNA isolation from cheek cells. This was a relatively short exercise, okay? And I divided the steps here into two parts. We have the one surrounded by the red rectangle and the one surrounded by the green rectangle. So can any one of you discuss or recall what was done in the first part?
Sa first part, sir, we extracted cheek cells from sa mouth yun, sir. And
Okay.
for this, kailangan ng falcon tube na may kato isulod dayon ang saline solution with katong tube na cheek cells from before, and then centrifuge din siya to pellet the cheek cells from the solution.
All right. Okay, so that is correct, no? Okay, so the cheek cells, the source of our cheek cells were from the inner linings of the mouth, so epithelial tissue, the inner linings of the mouth. And we got the cells through the, we gargled saline solution, so dapat 0.9% sodium chloride, so that the cells will also be preserved. They will not just burst, no, or shrivel. So after that, as you said, we now had the wash. We call it the wash. And then from the wash, we needed to put 1 ml at a time inside a microcentrifuge tube. And then we had to centrifuge that. And then what happens after centrifugation? What will we observe?
The pellet, sir.
All right. So the cells will be at the bottom of the tube as a pellet, and then we will have a supernatant. So our goal was to make the pellet the size of the head of a matchstick. So what we did in the video is that we discarded the supernatant. We added more wash now from the mouth, and then we centrifuged again so that the pellet, the cell pellet will increase in size. So the more sample there is, the better, more DNA. Right. So that was the first part. Now we have the pellet of cells, the cheek cells. What now happens in the second part? Anyone? Okay, see Kyle. Kyle, are you listening? Okay. What is now the next step after we got the cheek cells, the pellet of cheek cells? Or anyone who can recall.
Ano sir, kanang mag-undergo na sa PCR or polymerase chain reaction.
Not yet because this is still DNA isolation. Please recall, after we got the pellet of cheek cells, what then did we add? What is this chemical here?
Ay giad ang
Itong
instant
instantly,
sir.
Okay, sige, katong una sa naka-answer. Okay, the instagene matrix, no? You mentioned that. So, can you elaborate more? What then was done?
Gimix ang instagene matrix sa cheek cell pellet sir and gi
Incubate.
yes gi-incubate sir and then katong sa water bath and dry bath sir.
All right. So, there was incubation. There was the addition of the instagene matrix. So, the name instagene is not really a technical name, no, or a chemical name. It's actually a brand name. So, did any one of you research on what is in the instagene matrix? Or what does it do?
one, one, sir. Mabasahan ako, sir, kay yun na katong gina-read niya itong mga cofactors in the na present sa enzyme sa kanang sa cheek cells para in case na like the nato isa ka enzyme, sir, na kung present pa to siya kay mo disrupt sa DNA na naa sa sulod if ever nakapato.
All right, that's correct. So remember, after this exercise, we did not go directly to the next step. No, we refrigerated our sample. So in order for the sample to be preserved properly, the addition of the instagene matrix is important because, as you mentioned, it prevents the action of certain enzymes from disrupting the DNA or degrading the DNA. So it contains chemicals that will disrupt, particularly you mentioned the cofactors of that enzyme, so that that enzyme cannot do its work. Now, so we want it to be inactive, and that's the purpose of the instagene matrix, to preserve the DNA sample. So that's all in exercise 7. It's actually very straightforward, right? So I can imagine if you're the one doing this in the lab, you will finish in 30 minutes, di ba, in doing this. So DNA extraction or isolation is actually quite straightforward because we are now using special chemicals like the instagene matrix to do the job. No, hindi na siya mano-mano. Okay, so another question, what's the importance of the incubation? Why do we need to heat the sample?
So the nature some proteins
All
in the
right.
chick cell.
Yes, that is correct. So it will denature the proteins, particularly those that are part of the membrane from the cheek cell so that the DNA will be freed. No? Remember, the DNA is found in the nucleus of the cheek cell because that's a eukaryotic cell. So the nucleus should be broken. And one way to do that is to subject it to higher temperatures. So that's the importance of the incubation as well. Okay, so why, how is this related to the next exercise? Okay, to PCR. Okay, so imagine we have already extracted the DNA from the cheek cells. No, that's a human cheek cell. So the DNA in the human cheek cell is stored in 46 chromosomes. No, so these are the chromosome complements of a male diploid cell. No, X, Y, and N. So parang ganyan. So this is literally what we have extracted from our cheek cell sample, no, in the chromosomes. So, but there is one gene of interest that we want. Anyone from who are listening right now know what that gene is? We extracted DNA, but we are not interested in the whole DNA. There is just a particular gene portion that we want. What is that? Which chromosome is it found? You have 23 choices, 1 to 23.
16, sir.
All right. The gene that we are interested is in chromosome 16. Okay? So the DNA that I got from my cheek cells, it has chromosomes there and we are just interested with chromosome 16. Okay? So what is that particular gene? That gene is the ALU gene. Okay? The ALU gene is found in chromosome 16 and it's part of an intron. So if you haven't discussed DNA yet, no, in genetics, just to remind you what an intron is, it's an intervening sequence in the DNA that is not translated into a phenotype. No, so meaning to say, it's okay if you don't have it. So all of us, no, humans, we have the ALU gene in chromosome 16. And of course, the chromosome is in pairs because we are diploid. Okay, so what we see here in the illustration on the left is there are three scenarios. Now, for me, for example, because I extracted my DNA, one scenario is that in both of my chromosome 16, I will have the ALU gene. Okay, so meaning to say I am a plus plus for the ALU gene.
Excuse me,
Another
sir.
scenario, yes?
Sorry, sir, pero nawala ang imong slide, sir.
Oh, okay. Okay, I will reshare again. Okay na?
Yes, sir. Thank you,
All right.
sir.
Thank you also for telling me. So chromosome 16 is our target, and in chromosome 16 is the ALU gene. No, it's in an intron. And our goal, in my case, my own DNA, I want to know if I am homozygous positive, meaning to say if I'm a plus plus. So you can see here, both chromosomes have ALU, or if I'm a minus homozygous negative for alu, or if I'm a heterozygous, no, I am a plus negative for the alu gene. Now, if I happen to be negative for both, I don't have to worry because, as I mentioned, it's part of an intron, so I don't really lose anything if I don't have the alu gene. It just so happens that I could either be of these three scenarios: plus plus, minus minus, or plus minus. Okay, that's really the goal of our DNA extraction. No, so I want you to connect that to our next exercise. So in terms of genotype, these are the terms. I could be homozygous positive, I could be homozygous negative, or I could be heterozygous negative, a positive negative for the alu gene. Okay, so meaning to say, whatever we extracted na DNA, we don't need all of them. No, in fact, we are wasting our time if we're gonna analyze all of them because there's a lot. We only need to analyze one gene from chromosome 16. Now, my question to you is, how do we do that? Can anyone answer? We only need the gene from chromosome 16 that has the alu sequence. How do we do that? We have the whole entire genome. Any wild guesses? What's the next exercise again after DNA extraction?
PCR, sir.
The PCR. Okay. So, okay. So before I go to the relevance of my question, okay. So take note here. If I have homozygous plus plus, the gene that will be isolated will be longer. It will be 941 base pairs. But if I don't have the alu, the gene that will be isolated will be much shorter. It's 641 base pairs because it lacks the alu segment. Diba? This is logical. And if I have heterozygous condition, then one of my genes will be 941 base pairs long and the other will be 641 base pairs long. Okay, because one will have the alu and one will not have the alu. And the way we isolate this portion exactly, okay, and it's possible to do this, is through specific gene amplification. Specific because you only are working on a certain gene. Amplification because you're going to increase the number of that specific gene. And the process, as you mentioned, is called polymerase chain reaction, or PCR. Okay, so again, in exercise 7, we started with the extraction of DNA, and there are a lot of them. And then in exercise 8, we are trying to specify now just a specific gene in chromosome 16, the alu gene. And we are going to separate that from the rest, and we're going to multiply that many, many times. And that's done through the process of PCR. Okay, so what is PCR now? Okay, so PCR is actually a process that can produce many, many copies of a specific target gene. And our target gene is the alu gene in chromosome 16. And if you read the manual, there are three basic steps or cycles. No? First, the heating, and then there's the cooling, and then replication. So the technical term is this. First, there's heating or the denaturation. Okay, so if you remember your DNA replication in your general biology, DNA replication needs the two opposite strands of DNA to separate first. No, that's denaturation, and you can only do that by heating the sample to 94 degrees Celsius. And then after that, um, is the annealing portion. No, that's the cooling portion. Annealing, meaning to say, in order for the DNA to be replicated, a primer needs to attach first to the separated strands. And that can only be achieved at 60 degrees Celsius. And lastly, extension. So we are now going to replicate both strands. That's at 72 degrees Celsius, and that's the work of the DNA polymerase. Okay? So any questions as of this moment? Any clarifications? If you have questions, let's address them now before we continue.
None so far, sir.
All right, so I really hope that you understand the connection between exercise 7 and exercise 8. Okay, so that's very important. It might not be emphasized in the manual, but I'm saying it now. Okay, so notice here that in the video, it took 4 hours. Why is this only around 3 hours, sir? Okay, the answer to that is it takes some time to reach the temperature, di ba? If you looked at the video, the timer will not start unless, for example, 94 degrees Celsius is reached. So that's why there are some waiting time in between the steps. So we took a little bit more than 4 hours in the actual video. Okay, so this is going to be repeated 40 times in our case. Okay, and then after the 40 times repetition of denaturation, annealing, and extension, then we finish our PCR. Okay, so let's look at that in detail. So this is the DNA, for example, that we have isolated, and we are only going to target this blue colored gene. Now, this is the allogene in chromosome 16. Okay, so how do we do that one? Okay, so first step is denaturation, diba, at 94 degrees Celsius. So the two strands of the DNA, the two complementary strands will separate, no, unzip. And then annealing. So in the annealing, that's at 60 degrees Celsius. And that's where the primers, if you can remember what primers are, attached to the unzipped strands. Okay, let me ask you now, as early as now, how do you define a primer?
Sila ang start sa sequencer katong sa lagging strand.
Yes, lagging and the other strand. What's the other strand?
Leading strand.
So yes, you're correct. So both lagging and leading strands need primers. No? So in order to start the polymerization of a complementary strand. So another question, are the primers made of DNA or RNA?
RNA.
Yes, so you remembered the lesson, no? So RNA, the primers are made of RNA. And primers can be engineered specifically. No? So notice here that the primers, where did they start attaching? They started attaching at the opposite ends of your allo sequence, di ba? They did not attach somewhere else. They attached specifically at the ends of the allo sequence. And the reason for that is our primers are very specific. No, so there are you can actually customize your primers. No, and before you can do so, you need to know the sequence of DNA at both ends of your allogene. No, so sa atin, we already ordered that from a company, and they already gave us a pre-engineered primer that will really stick to the allogene ends. Okay, so that's not our problem anymore. So just that, not important. The primers are RNA strands, oligonucleotides, and they will stick just at the right ends of our gene of interest. In this case, the allogene. That's annealing. And then extension at 72 degrees Celsius. So it takes 2 minutes, a little bit longer. Who does the extension, as you see here? Or what enzyme does the extension?
Polymerase, sir.
Yes,
DNA
they're DNA polymerase. So you need to be specific because we also have an RNA polymerase. So this one is DNA polymerase. Okay. Is it a human DNA polymerase? Yes
most
or no? No. Why? Why can't we use a DNA
certainly
polymerase from humans? Come again?
this was specific, sir.
Not specific. Remember, the DNA is from me. I'm a human being. Okay, but then you answered that our DNA polymerase here is not from humans. You're correct. Why? Why should we do that?
Kay if sa human nga DNA polymerase kay ma-degrade siya kung taas na ang temperature.
All right. So remember, our extension temperature here is at 72 degrees Celsius. And remember, in the normal human body temperature, our highest will be around 37 degrees Celsius, di ba? So if we put a human DNA polymerase here, it will not work. It will be degraded. So you're correct. So it has to be a DNA polymerase from another organism that can withstand high temperatures. And the name of that polymerase is Taq polymerase. No, the Taq polymerase. What does Taq stand for? Clue, it's a bacterium. It's a thermophilic bacterium.
thermos
See,
aquaticus, sir.
that's correct. Thermus aquaticus. No? So there are actually other sources also. So if you read your manual, there are other, or if you research in the internet, there are other polymerases, and they are named after the scientific name of their source. So in our case, we are using Taq polymerase because it's from a bacterium that loves heat or hot environments. And the scientific name of the bacterium is Thermus aquaticus. No, so we are using Taq polymerase. Now, all of these are achieved, no, the denaturation, annealing extension, and then going back to denaturation. We do that automatically using a machine called the thermal cycler. Okay, so the thermal cycler, if you saw the video, you just need to program it, and then you put your sample and you can leave it. No, it will automatically change from 94 degrees Celsius to 60 to 72 and back again 40 times. No, so that's our thermal cycler. So it allows us to do PCR. So take note, PCR and thermal cycler are not the same. Thermal cycler is the machine, PCR is the process. Okay, please remember that one. So, of course, having the thermal cycler is not enough. No, remember we added that yellow substance first into our DNA sample. That yellow substance is called the master mix, if you can remember. And the components are these, the ones here on the left. What are these? Ano tong may 5 prime, 3 prime? 3 prime, 5 prime. What are these?
Strands
What
of
are
DNA,
these?
sir.
Ah, strands of DNA. What? What are they? I'm circling it now.
Primers,
Okay, so ito, so sa master mix, no, the illustration here on the left, you can see primers included. So there are primers. What else? What are these yellow oblongs? Ito, ano to?
not polymerase,
Taq polymerase should also be there. And what about these small dark blue T-shaped things?
Nucleotide, sir.
Nucleotide should also be present. And what ion needs to be present as well?
Magnesium, sir.
Magnesium ions. And of course, you need a buffer. So, meron ring buffer dyan. So, remember that the components of the master mix, you have the primers or the oligonucleotide primers. You have the nucleotides themselves, the A, T, and G, and C. And then you have the Taq polymerase. And then you have the magnesium ion. So question, what's the purpose of the magnesium ion in the master mix?
Cofactors, gyapon sir, para sa enzyme na i-catalyze ang reaction.
Yes, so magnesium ions are cofactors for the Taq polymerase. Now, they will help the Taq polymerase do their work. So that's why they need to be present. And therefore, there was a warning in your lab manual. When you transfer the DNA sample, make sure that no blank should be included. Do you remember that? Pag-transfer nyo dito sa preparation for PCR, you should not transfer the instagene matrix beads. Diba? Dapat wala. Why? Because the work of the beads is to prevent the cofactor from working with the enzyme. So our goal now is different. Our goal now is we want things to happen. In the previous exercise, we want to preserve the DNA. So now it's opposite, so there should be no instagene matrix present. So after the master mix, you then mix your DNA sample, and then you have your mixture now ready for PCR. And you put them in PCR tubes, no? Okay, so PCR tubes na dayon. And then run the thermal cycler. So it will run for several hours, and our target was 40 cycles. And notice here, not all of the products are allogenes. No, some of them are quite long. So you see here, there are gray extensions. But notice here at the bottom, there are these na wala ng gray extension. They are completely cut, clean cut. These are our targets. So these are the allogenes that we are targeting. And if we have 40 cycles, guess what? You will have 2 to the power of 40 of these allogenes, no? 1 times 10 to the power of 12 allogenes. So this is what we call as gene amplification, and that is the goal of your PCR. Diba? So from DNA isolation, you wanted a specific gene called the allogene in chromosome 16. You do PCR, you now have 1 times 10 to the 12 copies of your allogene, not of the whole DNA, not of the entire DNA. So that is quite amazing. And these can be done as one workflow in the laboratory, no? But it was just divided in your laboratory exercises. So the next laboratory exercise is actually a continuation of this workflow. No, it's called agarose gel electrophoresis. And the results of our PCR, we're going to load in an agarose gel. And just like in SDS stage, we are going to run the gel in an electrophoresis chamber. And we can see results, the bands of DNA forming on the gel. So that will be on the next exercise. Okay, so I hope you understood the importance of the connection between PCR and DNA extraction. No, that's very important. And of course, what we did in the lab is purely for demonstration. So your big task actually, especially in the pre-final output, you need to have a specific experiment. Maybe you can research from a journal wherein they use DNA extraction and then they use PCR and then agarose gel electrophoresis in their workflow. So that will be, I will tell you about that requirement in detail in the next video teleconferences. So for now, your task is to finish your exercise number 8, okay, because the deadline is this Saturday, right? So that's everything that I wanted to present in this video teleconference. Are there any questions or concerns?
None for me, sir.
All right. So nasabtan ra? You were able to understand what I explained?
Yes, sir.
Yes, sir.
Okay. So recording of this video teleconference will be posted after this, so you can review on that, and your classmates can also view what we have talked about here. Okay, so thank you very much, guys, for joining in this video teleconference. No, so always strive to join. Otherwise, if there's no one, I will not do a video teleconference, and I will not be able to record what I want to say. And I believe what we are discussing here is important for everyone. No, so walang video teleconference, walang recording that you can look back on. So always attend video teleconferences. So please tell your classmates that. All right. So again, thank you very much, and I hope you'll have a productive day ahead. Okay, and see you in the next video teleconference. You are now dismissed.
Thank you,
Thank
sir.
you, sir.
Goodbye,
Yes,
sir.
sir. Yes, sir.
You're welcome.